Let's go over your questions during the break. One question was is the Luca the ancestor from a cell? The simple answer is a yes, right? So Luca, by definition, is the ancestor form of the first ancestor of all life forms. And since we consider the first life form must have been a single cell, in particular the prokaryotic cell. So in the cell's ancestral form, we can easily relate and point to Luca is the logical guess. And if you are kind of confused about this endomembrane system, uh, the reason why this eukaryotic, some of at least majority, in fact, a majority of the cellular organelles of eukaryotic cells belong to this so-called endomembrane system. The reason why is the origin first. Their origins are all considered to be originated from the cell membrane. That's why it's called the endomembrane, which is equivalent to the external outside the cell membrane. But the only difference is this endomembrane system is internalized membrane, like which are originally same material as the cell membrane. So you do have this nucleus, which we think that originated from the cell membrane, and also attached and continues with this nuclear membrane, which is ER, endoplasmic reticulum. Some of these ERs, they have ribosomes attached on the surface, then we call that as a rough ER. Some other endoplasmic reticulums are smooth because they don't have attached ribosome because of their different functions, and a little distantly located is a stack of pancake-like Golgi, and we think that even these different functioning Golgis are also derived from this invagination inside the pushing, inside the folding of this cell plasma membrane during the course of this evolution, and some isolated small structure called lysosome. We do have many, to name a few. Okay. So these are the endomembrane system, and once again, the reason why all these different organelles functioning differently are all like classified into one category of endomembrane system is we believe they all derived from the same origin, which is a cell membrane. That's it. And are there any organelles that do not belong to this so-called endomembrane system? Yes, notable such exceptions are mitochondria and chloroplast, which we do not even have, are the two very distinct exceptions, which we cannot include them into this so-called endomembrane system because their origin are totally different, as it turned out. It is interesting that Golgi recognized the zip code of a protein and carry them properly. Yes, I agree. It's very interesting and impressive that Golgi as a one organelle can recognize different so-called signal zip code with which different protein are being translocated to different area. And the secret of this recognition is in that, I mean, guess what? How the Golgi can distinguish and recognize different signal signs of so-called zip code and then deliver different zip code bearing cargos to different location. It's all receptor protein. Different receptor protein that can bind or recognize a different signal as a zip code. And it's just all matter of now, if Golgi can have different types of such receptor protein that can recognize different signal, and the only other problem that you need as a Golgi, you need to solve is distribute these different type of receptor protein in different area, and then you ship different part of this to a different location. That's how they handle this recognition and delivery. All cells come from other cells through a process of cell division, but I didn't understand about what exactly the cell division is. Okay? You don't have to have any frustration with that. And then good question. If you are not comfortable about this cell division, yeah, so what? Cell division is one cell become two cell, and this is the means by which we proliferate and we maintain the longevity. Yeah, we can live forever, as I put this in the first lecture of this course. But how does this cell? There are two components of the cell division, and for any cell that needs to carry out this cell division successfully. First is you have to replicate your genome. So if you had, let's say, if you had 46 chromosome originally, then temporarily you have to double up this chromosome into 92, double the amount of chromosome, very temporarily. So then split into two. So each so-called new cell, daughter cell we call, inherit the exact number of 46 chromosomes. So this DNA replication, so that you duplicate the chromosome number, is extremely critical and extremely very important preparatory step for any cell division. And the next thing that you need to worry about is physical separation of your cell surface, which is, as we went over in the first session, that you need to squeeze in the case of animal cell, on the center area, so that this area can separate into two things. So any cell division is two different parts of a procedure. One is your chromosome duplication, so that exact same number of original number of chromosomes can be transferred into two cells. That's one part. And another thing is a physical separation, severing all this your cell surface into two. That's how you do it. But that's the mode of cell division for your body cell, normal body cell. And this way of one cell become two cell like this, we call as a mitosis. So this is a typical cell division through which one becomes two. But the special type of cell division, which one cell continue to divide to become one original cell become 4 different cells, is we call meiosis. And this special type of cell division, meiosis, is only for gametes. What's gamete? Sex cells, sperm and eggs. And this gamete production, there is a specific reason why you cannot stop. You cannot stop at this point, just like the normal, so to speak, normal cell division, which is mitosis. But you have to go further, one step further to become 4. There is a reason why. I will just briefly say the reason instead of going into actual real details, because that's something at this point unnecessary. Although I am going to handle this specific reason later on because that's important. But so this gamete production, which is meiosis, the reason why this one cell has to become 4 cells as a result is because you need to reduce the amount of genome, whatever it is, whether it's a chromosome number wise or the total amount of your DNA, you have to reduce them into half, 50%. Why? Because the purpose of this cell division is to produce the gametes. The original parental cell was a haploid cell, but from diploid cell, you should provide haploid cells. That's why you cannot stop over here, but you have to go one step further. Okay. The detailed reason why to reduce your integrity into the half, which is haploid, then you need to go through one more round of cell division. I will not talk about right now because it's maybe a little more kind of confusing, but that's the reason. Can modern mitochondria engulfed in eukaryotic cell is viewed as a kind of a cell? What do you think? If this cell, like mitochondria, if we can consider them as a still kind of cell, then outside of the so-called host cell, those mitochondria should be able to survive. In other words, if you isolate mitochondria from the host cell, then let them live independently, then can this mitochondria still duplicate their genome and then divide? If they can show, then we can consider them as a kind of a cell. If not, now maybe the ancestral form of this mitochondria must have been independent, a kind of a cell, but not anymore because they during these course, long period of time, they must have lost such the autonomy, independency. Okay, so that's the kind of easy distinction. Whether can they survive outside or can they not survive outside? I like this is very cute cartoon. Love it. So I wanted to share this cartoon with you. No one is paying any attention though. Uh, how do prokaryotes get energy without mitochondria? It's a very good question, actually, because mitochondria. I said mitochondria is the one that responsible for producing ATP, but that's only the part of the story. Maybe if I'm drawing a cell, let's say this is a mitochondria. For simplicity, I omitted all other structures like a nucleus or anything like that. And So this mitochondria is the one generating huge amount of ATPs, a lot of ATPs. But for that, you need to provide mitochondria with oxygen. So that's the thing. But that's only the small a part of the story. Although the mitochondria can generate so much amount of ATP, but without mitochondria, still a cell can manage to produce a very small bits of amount of ATP in the cytoplasm. So let's say glucose on exemplary food material, glucose enter the cell, then this glucose will be split into two. Okay. During the procedure, small amount of the ATP can be generated, although that amount of ATP is not really nearly enough for this eukaryotic cell used to survive, maintain their lives. But for prokaryotes, this small amount that can be produced outside of this mitochondria may be sufficient for them to survive because of prokaryotes, their energy demand is very small. Okay, that's one reason. So without mitochondria, prokaryote can somehow manage it by utilizing this early part of this metabolism. This is a early part, and then eventually all this metabolism will be connected into the mitochondrial metabolism. And the second more important reason is a lot of bacteria also have developed that the reason why this mitochondria can produce this so much amount of ATP is because of some very specific enzymes lined up inside of inner membrane of the mitochondria. But then cleverly, the bacteria transport these enzymes in their cell membrane. So without mitochondria, they can similarly function this important metabolism which is being carried out in the mitochondria. So even those bacteria can produce as much amount of ATP as this eukaryotic cells. That's another very good reason. Yeah, I'm not sure how many of you can actually follow these things, but that's the answer actually. How the prokaryote can survive without this mitochondria? Is there any other kinds of a protein contrast with glycoprotein? Interesting question. Is okay. What do you want? I mean, so like you want to see some other version of this naming, like so. Glycoprotein means protein having carbohydrate, right? So what else do you? What kind of other combination do you want? Like so, how about this? Lipoprotein. Would that be enough? So lipoprotein is what? Protein having lipid. That could be some kind of the example, contrast with glycoprotein. Protein having carbohydrate, but this lipoprotein is protein having lipids. Yeah, this is something the real existing one, lipoprotein. Actually, yeah, this should be very familiar. LDL or HDL. This good cholesterol and then bad cholesterol. These are examples of such lipoprotein. Okay? Proteins having a lot of lipid inside, kind of. And we do have another many different examples of such lipoproteins too. Someone brought up this, so I just look it up. So this is manga. Actually, she is an international student. There was a manga name. The title of this manga is "Cells at Work," and this is a Japanese manga, the cartoon. So I look it up, and then actually Wikipedia has this particular information. So if you type in Google if you Google in the cells at work cartoon, then you can specifically find this version. And it looks like all different kind of cells in the human body, including different types of white blood cells and red blood cells and different kind of cells. Probably the author described their function, roles, like in different metaphor of a human body. So although I didn't bother to pick up and try to look at it, but if any one of you are interested in learning in interesting way, in fun way of different roles of different cells, probably you might want to actually check them out. Thank you. Okay. So exactly that's about time to start the second session. What a convenience. Okay, good. So we do have outside of the cell, we do have so many different things. Collectively, we call that as an extracellular matrix. Um, so this so-called extracellular matrix include so many different types of proteins, famous proteins. And okay, one question is, if this so-called extracellular matrix, which are all different types of proteins, what do you think? Are these all glycoproteins? Would all extracellular matrix protein be glycoproteins? My question is. Then answer is yes or no. What do you think? Come on, what do you think? Would all extracellular matrix protein be glycoproteins, or only some of them be glycoprotein? What do you think? Answer is all the extracellular matrix protein would be glycoproteins. Why? Extracellular matrix proteins are what? So this is a cell, and this is a cell. So we are all living together as a multicellular protein. And extracellular matrix proteins are where? Where are they? They are all over here. Outside the space, extracellular space is the area where you can find this, whatever, ECM protein, extracellular matrix proteins. And where did they come from? So inside of the cell, they originally were made inside of the cell and they were shipped out of the cell through the procedure of what? Exocytosis. Then, if there was a case, all these, so these are the result of a secretion. All these secreted proteins will have the same fate, starting from what? Raphael, going to the Golgi, and then go to the cell outside of the cell. So during those two places, they must have obtained heavy glycosylation, so they must be all glycoproteins. It's just kind of like trying to have fun to give you this question to remind you what you have learned in the previous week. But more importantly, can you name one very famous example of extracellular matrix protein? Although there are several different types of extracellular matrix protein, but if we can remember exemplary extracellular matrix protein, then perhaps we can relate and understand why they are here outside of the cell. Can anyone already know? Does anyone who already know that extracellular matrix, one example of extracellular matrix protein? It's very famous protein. The what? white blood cell. White blood cell is a cell itself. So extracellular matrix is a protein. So that's just a difference. And this particular protein, we know that they are so abundant in the pig skin, so everybody loves to go to have a feast of a pig skin because of this. Yeah, collagen. Collagen is one very exemplary extracellular matrix protein. Why they are outside of the cell? Because collagen is very flexible protein, just like spring-like protein. Collagen is another such a fibrous protein. It provides a spring-like function. So it provides a cushion. That's the reason why they are outside, so that they are fillers, like a stuffed animal's fillers. So being present outside so that they can provide the flexibility and protections. And that's why on your skin, if you are still have a plenty of abundant collagen, your skin look very fresh and young. And as you grow older, then you tend to lose the content of collagen, then your skin now develop more and more wrinkles. So that's the relationship. Okay. So that's only one. And similarly, so many other such proteins function outside by providing very similar and some other important functions. All together make up the extracellular matrix. Like this. That's all. So collagen, proteoglycan, fibronectin, whatever they are all outside, and they all highly contain high contents of carbohydrates, so high glycoprotein. That's it. Okay, so that's just kind of brief consideration of extracellular matrix protein. No further details information we need to worry about. So now we can move on to observing different forms of cells. Plant cell, yeah, everybody knows. Plant cells, the different structurally different feature of a cell, plant cell, three different features. First, cell wall, rigid outside protection, and chloroplast, which provide the photosynthesis function. And another, probably central vacuole, is probably you did not really hear this about as frequently as others. So the central vacuole is internal sac-like things, which in animal cell we don't really see this big central so-called vacuole. Vacuole is a big empty sac. We call it a vacuole. Although animal cell may have a small vacuoles inside of cell, maybe, but we do not have such a one big major central big vacuole. One thing is plant cell tends to have, as they grow older, this central vacuole tends to become larger and larger and larger. Why? Because plant cell use this central vacuole as a big storage place, okay, for their important materials as well as their waste products. So as the cells grow older and older, they must accumulate more and more their particular metabolic product as well as particular waste products. That's why older cell will have a bigger, larger central vacuole. And different plants will have their own unique particular type of material, chemicals stored in their vacuole. Like, for example, tobacco, right? Tobacco plant is renowned for having particular compound, which is nicotine, right? Not every plant produce nicotine, but only some specific tobacco plant are able to produce the nicotine. And this nicotine will be stored in this each cell's central vacuole. Okay? And toxic, did you have a question? No, okay. Hey, just by waving, right? Uh, the milkweed, if you still remember, the milkweed plant producing the toxin, yeah, that toxin will be stored in the central vacuole of that plant. So this vacuole is their general common storage place for whatever the chemical they value. Primary cell chemical composition, who cares? Don't worry about it. Secondary cell wall, don't worry about it. Yeah, we can move on to the prokaryote. Briefly, let's take a look at briefly prokaryotes and then come back to the eukaryotes. So prokaryotes are all like those cells not having nucleus and organelles, but there are two different groups of prokaryotes, probably perhaps you know by now. One is called archaebacteria. Like sounds like ancient, very ancient bacteria, archaebacteria, whereas the real bacteria, maybe a modern bacteria. So scientists distinguish these two by calling differently, like archaebacteria versus eubacteria. Eubacteria means true bacteria, right? That doesn't mean that archaebacteria are not true, untrue bacteria, but anyway. And the interesting thing is actually we, as I have mentioned several times, the eukaryotic cells actually evolved from specific branch of archaebacteria. So we are, as a eukaryotic cell, we are evolutionarily closer to the archaebacteria than the eubacteria. So it it can be applied to both eubacteria as well as archaebacteria, but there are so many different, tons of different types of bacteria, including eubacteria and archaebacteria, present these days. But no matter how many different bacteria are there, basically, if we want to classify them by their outside appearance, there are basically three different forms. Hello. One is round ball-like shape, we call as a coccus. Have you ever heard of Staphylococcus, which is very deadly bacteria? Staphylococcus. Or how about so you sure you have not heard anything like this before? This one? You have? Yeah, this is Staphylococcus. So this coccus means ball-like. So if you examine this Staphylococcus under the microscope, each bacteria look like grape. That's why it's called 포도상 구균, grape-like, raw ball-like bacteria. That's what this staphylococcus. So here, what we can take is, oh, coccus means any bacteria look like this ball-like individual bacteria we call coccus. On the other hand, bacillus is like a pill-like shape, a little round, rod-like, rod-shaped bacteria called bacillus. One very famous such bacillus form of bacteria is E. coli. E. coli is the which reside in our intestine. So this is nothing to do with any particular evolutionary different types or anything like, but simply strictly we distinguish by appearance. Two differences. And also not as common as two different coccus or bacillus, but another form like spiral like shape or rather long spiral type of shape of bacteria we call spirochete. Okay? So these three different types of bacteria, either coccus, bacillus, or spirochetes are the three standard different types of bacteria. And out of each different form, a little variation. Like coccus can be a little variation forms, then we call as a coccobacillus type. But these are just, you don't really have to worry about. These are the first prototype typical standard form, and in some other a little more advanced form of a cell biology textbook, they have a further distinctions of like coccobacillus or vibrio or spirillum, kind of a little bit of a variation form of this, the first standard form of bacteria too. Now, so this is a rod-shaped bacillus, typical bacillus form of bacteria, and common standard features of any typical bacteria are presented here. So, for example, cell membrane, also known as a plasma membrane. Cell membrane is there, just like all other type of cells. It has to have a cell membrane somewhere. They do have a chromosome, bacterial chromosome, but unlike eukaryotic cell, they do not have any nucleus. It's just so-called naked, without any protection. It has that's what what makes this cell as a prokaryote, obviously. And outside of the plasma membrane, cell membrane, any typical bacterial cell has a cell wall, their own version of a cell wall outside of the cell membrane, that's it. So they can enjoy extra protection, fine. And obviously, they also must have a prokaryotic ribosome, fine. But what about this capsule? Capsule is present even outside of cell wall. So the capsule is even covering the most external protective layer, which is a cell wall. So this capsule is actually an optional thing. Not every bacteria have this capsule. Capsule is something like a sugar coating. It's made of carbohydrate. Capsule is made of carbohydrate, and this is a kind of optional thing. So not every bacteria can have a capsule. Only some of bacteria can have a capsule. But if you, as a bacteria, if you have capsule, and it provides some extra characteristics of, like any bacteria having capsule is more virulent, more effective in infection. We call it virulent. So, like if we were invaded by two different forms of bacteria, one is having capsule, another one is not having capsule, and we worry much less about those who do not have any capsule. We take more serious about defending against those who have capsule. Of course, in these days, we are blessed with whole bunch of different variety of what antibiotics. So without having capsule or not, we just kill them all using by dumping antibiotics. Will be very easy. But just in case we do not have such the convenience of antibiotics, then those who possess the capsule, then we have much more difficulty in defending against them. Why? Because without any antibiotics, what's our alternative resource with which we can defend? What do we have? If we do not have any antibiotics, with what can we fight against those invading bacteria? Anybody? We do not have any antibiotics, then we get killed by bacteria? Or is there any do we have still hope that even without any antibiotics we can somehow win over bacteria? If that's the case, what do we have? White blood cell. Yeah, we do have our own defense called what? Immune system. Immune systems like the main frame is made from our whole variety of white blood system. And now, can you remember that among those, one particular form of white blood cell who can eat invading bacteria? One particular white blood cell can capture those bacteria and eat them, right? Yeah, that's what we can rely on, even in the case we do not have any antibiotics. But in such cases, this capsule will become very interfering one for our efficient immune response of capturing and eating those bacteria. Why? Because this capsule will make the surface of this bacteria very slippery. So this white blood cell try to grab this, and oops, they just drop. Just like everybody have tried to pick those what was that? Whatchamacallit? The little dolls and those stuffed animals and the booth. Okay, and then you do not really have very luck in succeeding one because you always lose the grip, just like that. So this capsule will make such a grip so slippery, and that's how they can escape such immune response. And another thing is there are two different types of hairs. One long hairs and very short hairs. And we do have different names. Whatever they are, both short hair and long hair, in general, they are used in their mobility. So this long hair-like things provide some propeller-like function. Okay, we call it the flagella. They are made of proteins. And the short hair-like things, we call that what? What's the name of this short hair? Can you pronounce this? How would you pronounce this? This. Pili, which is plural. So singular is called pilus. And plural, multiple forms, it's called pili. Okay. So this pilus is the one, the short hair, also provides some of the mobility as well as some other additional functions like for example, what is this? Genetic recombination? What is this? Genetic recombination. Genetic recombination is the exclusive, the feature which is exclusive to eukaryotes. Like genetic recombination can be achieved through sexual reproduction. The asexual reproduction, which is being carried out by all prokaryotes, are not supposed to result in any such genetic recombination. Why? Through genetic recombination, you can achieve some genetic variation, but asexual reproduction will, they are supposed to produce only the clones, exact copy of your parental form. Then how can you obtain these characteristics of what? Genetic recombination? That's not fair. You are prokaryotes. And the reason, guess what? The reason why all these prokaryotes still wants to engage in only this asexual reproduction is what? What's the advantage of asexual reproduction? Can you think of any the kind of a couple of or maybe couple of or three at most? There are different advantages, benefit of having this asexual reproduction. Yeah, it's like high rate of replication means it's very fast. They can rapidly divide, like typical bacterial cell on the very good condition, favorable condition, they can even divide within 20 minutes. Oh my gosh. 20 minutes they can produce 2, and another 20 minutes they can produce 4, like they multiply so fast. It's very good, such a very strong advantage. So it's very fast, and also what? It's very cheap. You don't really need to invest too much resources. It's very efficient. So asexual reproduction is very efficient, so that the chance of success as an individual, if you like reproduce through asexual reproduction, chance of success is very high. Okay? That's why all these prokaryotes love to reproduce through asexual reproduction. But what is the very critical deadly disadvantage, which is a potential disadvantage of asexual reproduction? When you die all together. Everybody go to hell together. Not a single survivor, so to speak. Because you are clone. No matter how many are present in this small room, if you're a prokaryote, then you are genetically identical because you are the result of this asexual reproduction. So if things changes, now you prosper because of the particular favorable environmental condition, but environment changes so rapidly. So once this environment changes into unfavorable one, then everybody will respond to the same way. So everybody will die miserably, right? So that's the reason probably a lot of those eukaryotes, even though it is highly inefficient in terms of a time wise and in terms of the energy spending wise, but still they want to engage in this through sexual reproduction is because of this. They can generate diversity through this genetic recombination during this process of sexual reproduction, so that even those drastic environmental change, at least some rare few exception of this your genetic makeup has some chance of a survival, and that's how we managed to survive. They know, perhaps these prokaryotes, they know they are really deadly Achilles heel. That's why, on occasion, they have this practice of genetic recombination. Although it is only very small local instances, but they do have this. And through these pilots, they make this sexual, so to speak, very sexual reproduction like genetic recombination possible. How? This. Yeah. Are there any? Oh, here. This is a real photograph of two bacteria engaged in such recombination, genetic recombination through this. This is a long thread-like pilus. When they use this particular pilus in this recombination, we call what? Sex pilus, because they are being used through this exchange of their genes. So, hey, I have some genes. Maybe you might be interested in. Would you like to use my gene? And if this guy says yes, okay, then I will send you, okay, transfer these things. And then they accept. Even sometimes three of them engage at the same time, have this exchange of the genes. This particular type of recombination we call that as a conjugation. Through these sex pilots exchanging their genetic information is what we call the bacterial conjugation. But once again, even if you forgot this particular term, conjugation, yeah, it's not serious. Doesn't matter. No big deal. Okay? But simply, if you somehow wonder that, what do you call this? Yeah, there is a specific name for this conjugation. But you forgot, who cares? No big deal. So anyway, then what is being exchanged is the more important question. During this conjugation, do you have this exchange of your entire bacterial chromosome? No, it is not. This is another part. This is another very interesting feature of any bacteria, the genome, in that they have their main chromosome here, bacterial chromosome, which is circular, yeah, round circular chromosome. But in addition to, wow, why do I have this? By just moving around here, I achieve this activity, exercise, ring. Okay, good. Small, this is called the plasmid. So both bacteria and archaea have very small, we call it the extra chromosomal DNA. So you do have a main chromosome, but in addition to the main chromosome, you do have additional small, very similar DNA called plasmid. That's the definition of the plasmid. And the beauty of this plasmid is did I mention about the plasmid before here in this classroom? Did I? No, no? Okay. So let's say this is bacterial cell. You do have main chromosome, which is circular. But additionally, you do have small circular extra chromosome, which is independent from the bacterial chromosome, small circular DNA called the plasmid. This is a plasmid. The beauty of this plasmid is independent. They can replicate without any permission of this main chromosome. So in other words, they can increase the copy number. Although bacterial main chromosome can have only one copy per cell, okay, but plasmid can exist in multiple copies, just very similar to mitochondria. But in this case, not only few or several hundred, but even several thousand, something like, although it is an engineered version, but maximum, this same copy of plasmid, particular version copy, can be increased up to 5,000 copies with just a little trick. So single bacteria cell can have maximum 5,000 copies of same plasmid. Why do I bring up this? Because let's say you isolate one plasmid. Let's say you took it out one plasmid over here. You do have one plasmid. Okay? Now, this is a DNA. And can you imagine what if this is DNA. What if you put a fragment of human gene, for example, over here to become integral part of this plasmid? Is it possible? So what kind of human gene do you want to try on? Hey, how about I'd like to have insulin, human insulin gene. I want to take out human insulin gene and put it into a plasmid. Okay, is it possible? Yeah, let's try it. And then once I have succeeded in, let's put this back into original bacterial host. Now inside of this bacterial host, now you have this plasmid plus human insulin, and this can go up to 5,000 copy of this thing. Now inside of this bacteria, what's the fate of this human insulin gene? It will be regarded as same bacterial gene, and then will be treated as such, so that this human insulin gene will be what? Expressed. What do you mean by expression? Human insulin protein will be made inside of the bacteria. That was in 1975. It was the first ever trial instance of genetic engineering that we know of by now. That was the first case of genetic engineering that scientists at Harvard and Berkeley thought about it. Hey, would it be possible? Hey, there is a very interesting feature of bacteria. Have you heard of this plasmid? Yeah, I know. So if we isolate this plasmid and then put this human insulin gene, would it work? Let's try on. And they did, and it worked. So now we were able to produce insulin in the bacterial cell. Before this 1975 trial, how do you think we produced insulin? to treat the diabetics. Pig, the whatchamacallit, that was a pancreas. Yeah, pig's pancreas. You isolate pig's pancreas and then purify the small amount of insulin from such pig. Then obviously you would understand that the amount would not be enough. So that's why at the time the insulin price of insulin must be very, very, very high, pricey, right? But now you change the technique like this with the help of genetic engineering. Now the amount of insulin that you can produce is what? Each single bacterial cell can divide in 20 minutes. And in small flask, how many bacteria do you know how many bacteria live in a small volume of a liquid flask? Yeah, billions, right? So each cell can have up to 5,000 copy of this human insulin gene. That's why these days, have you ever tried this insulin treatment? No one. Obviously good. Nobody is having this diabetes now, at least for now, good. So, but the insulin is very, 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 very cheap. So that's how so the plasmid was the tool with which we developed this modern current technology of those genetic engineering. Although now it became already such an ancient techniques, especially in these days, like the gene editing era of gene editing using whole different tools. Like, have you ever heard of spelling seizures? Is it S or whatever? This genetic seizures called CRISPR. This is whole new technology that make all this ancient genetic engineering recombinant technology obsolete. But anyway, still historically and still nowadays, this plasmid is being used as a really reliable workhorse for all these different techniques of gene editing, gene manipulation. Okay, so that's the one good part of the utility of plasmid. Okay. Now, good news first and bad news follows. And this plasmid is the culprit of the antibiotics resistance spread. Nowadays, we have a big problem of so-called superbacteria. The bacteria showing resistance of whole multiple different kinds of antibiotics, as by definition we call superbacteria. Once you are infected with some kind of superbacteria, then there are no cures because no currently available antibiotics will work on such resistance bacteria. So now that's the time. Now is the time. Then your only hope is your immune system, antibiotics. But usually those those people who are infected with the superbacteria, usually that happens in the hospital because that's the place where so many different types of superbacteria are available. So whenever you visit the hospital, especially the general hospital, you should be very careful because that's where so many different types of superbacteria are around. Okay. So those people who already had to visit hospital are already kind of like immunologically challenged. So that's why they have a big problem over there. But here, why the plasmid is responsible for this antibiotics resistance. It's because that conjugation, this conjugation, during this conjugation, plasmid is the one move. Okay. Through this sex pilus, this plasmid will be transferred into the next bacteria, like this. So this plasmid is being replicated, but this newly synthesized replicated copy will move to this another bacteria instead of staying over here. So that's one of the main mechanisms by which this gene is being transferred from one bacteria to another. There is another kind of more complex mechanism with which actually the part of the chromosome can also be transferred onto the, another one, but I will deliberately avoid to discuss that. Simply just focus on plasmid transfer through this conjugation. But the problem with this plasmid is somehow for some reason we do not fully understand. Usually this plasmid tends to carry several different the genes responsible for this antibiotic resistance. So here on this particular plasmid, zoomed in, is a ABC or RSE, resistance gene A, B, C, all these three different genes responsible for different forms of antibiotics resistance. So here, antibiotics A, several different types of antibiotics will become obsolete in the presence of these particular genes. So here we have to review ourselves that what is the nature of antibiotics resistance? All these antibiotic resistances are the result of mutation. Original, you have a gene. Let's try to compare this scenario in previous this cartoon that you have already seen today. Like each different antibiotics are being developed in the hope of targeting specific the enzyme usually. So let's consider this scenario. This particular antibiotics, which is supposed to inhibit this bacterial DNA replicating enzyme, right? But then if some mutation occur in the gene for this DNA replicating enzyme, so that structurally this has been changed, so then now this antibiotics no longer active on this mutated version of enzyme. Then as far as this particular antibiotics concerned, this bacteria obtained resistance against this drug, antibiotics. Okay? Why? Due to this mutation in their original enzyme. That's how you obtain the resistance, antibiotics resistance. So all these antibiotic resistance are the result of particular, some specific genes mutation. So having said that, this is actually a real life example. Like all these pill-like cartoons represent antibiotics, the drug. So for example, one such mechanism would be antibiotics will have to enter the cell, but somehow you have a mutation in the protein, mutation in the gene making this transporter protein. So somehow this transporter becomes super transporter. So any antibiotics coming in can be picked up and then dumped out. Then these antibiotics have no effect because they cannot. Virtually, they come in and then they are all kicked out because of the mutation in this particular the protein. That's one possibility. And actually, it was like real instance that has been observed. Another, let's say this antibiotics used to target this particular enzyme. But somehow, the structure of this enzyme has been altered due to a mutation. So now, this drug cannot affect the function of this particular enzyme because of this mutation. Then there's no effect, become obsolete. Similar things, and some occasions even the mutation will create some new form of enzyme. So the old form of enzyme is still being affected by the antibiotics, but this new form of enzyme can bypass this. Hey, have a good time. I will go this. And some other times even they create whole new non-existing enzyme which can actively destroy this. Actually, this is the case for the antibiotics, the penicillin resistance. The enzyme, mutated version of enzyme, will actively destroy this penicillin. Okay, got confused? So many different occasions. So do I have to memorize all these different occasions? No, of course not. What's the point of this diagram that I am trying to show? What's the common scenario here? You have some specific way of inhibiting particular process of metabolism of the bacteria. That's how the antibiotics works. But somehow they can escape because of this mutation of that particular the genes making that particular enzyme, the metabolic process enzyme. That's all the common characteristics of all this the resistance mechanism. Once again, the key word is mutation, making this function of antibiotics obsolete, not working, is how this antibiotics resistance work. But so up until now, we have only just witnessed that the actual scientifically speaking, the mechanism of antibiotics resistance spread. But in real life, the main reason why these days there are so many different instances of antibiotics resistance super bacteria are so abundant is because of our stupidity, human stupidity, not because of any mutation or anything. Because even there are some occasions of antibiotics resistant superbacteria occurrence, but those bacteria are what? Basically the result of multiple mutations. The mutants, they're mutant. Regular, so like regular bacteria are. Regular bacteria do not carry any these mutations, can grow much faster than those mutant superbacteria. So if we do nothing and then leave them together in the mixed environment, then so to speak, the non-resistant, the regular bacteria will outcompete those superbacteria. But what do? We are selecting. We are like playing some kind of a natural selection using the antibiotics. You are pouring unnecessarily, dumping excess amount of antibiotics in every different occasions. Like even you go to the doctor's office for your common cold, which is caused by virus. So the antibiotics will not even work at all. But doctor prescribes usually the antibiotics for the precautionary reasons. So what's the end result? All those antibiotics will selectively kill those who do not have any resistance. So like selecting for those resistant bacteria. That's how we have so many frequent occasions of superbacteria in these days. Okay. Uh, yeah, it's a shame that I'm moving too slow. So I haven't reached even archaebacteria discussions. So I'd love to, as much as I love to continue on the discussion of archaebacteria, but it's already hit the 40. I don't want to ruin the rest of the Friday evening, so probably I have to stop here today. So have a nice weekend, and we will continue on the rest of the stuff maybe next